Hi everyone. Today we're going to be talking about cell communication, or Campbell Biology chapter 11. And if you're taking AP Bio, it's a part of Unit 3. And we'll just dive right into it. Okay, so these are the main types of signaling in cells. So there's paracrine signaling. This is a type of local signaling where the signaling molecules only travel like a short distance to neighboring cells. And then there's autocrine signaling, which is when the cell transmits signaling molecules back onto itself. So it's kind of like the cell just signals to itself using its own molecules. And then there's synaptic signaling. This is prevalent in the neurons and neuronal system. So synaptic communication can be both chemical and electrical. In the case of chemical signaling, they usually use neurotransmitters, whereas electrical signaling depends on voltage. And then finally, there's endocrine signaling, which is when hormones or other signaling molecules are secreted into the blood. And then through the blood, the signaling molecules can travel like long distances throughout the body. Now you might be wondering, we have endocrine, but where does exocrine signaling come into play? Now endocrine means that the molecule travels through the blood or another intracellular fluid such as lymph. However, exocrine signaling depends on secretions of signaling molecules via ducts. Now, these ducts can be like mammary glands, sweat glands, etc. So there's 3 types of secretions. There's merocrine secretion, apocrine secretion, and holocrine secretion. Now, merocrine secretion is when the signaling molecules are enclosed in secretory vesicles, and then they're secreted when the vesicles fuse with the plasma membrane, and as we learned in previous units, through exocytosis. Now, there's a lot that goes on during exocytosis, such as the formation of the snare complex, but we do not need to go into that much detail. And then apocrine signaling is when part of the cell is actually pinched off, and then the signaling molecules are incorporated in that section that's pinched off. And in holocrine secretion, it's when the cell actually dies and becomes the secretory product. So now we have a local signaling. Local signaling is basically all types of signaling besides endocrine and exocrine signaling. So we have gap junctions. These are a type of cell junction in the animal body. Now, gap junctions are crucial in paracrine signaling, like the nearby local signaling. So in gap junctions, there's the adjacent plasma membranes, as you can see in the diagram here. And actually, small molecules can diffuse between the connexins, which are proteins embedded between the plasma membranes. So they act as like a bridge. And these gap junctions can actually regulate what passes through these connexins using various mechanisms. So plasmodesmata are like little pores between adjacent plant cells. It allows for the continuous exchange of cytoplasm between plant cells. So basically, one plant cell kind of shares its cytoplasm almost with the one next to it. Now, plasmodesmata are important in the symplastic routes, which we will learn further in chapters, I believe, 30-something, 35, 37. But yes, we will learn more about that. But for now, just remember that signaling molecules can travel between plants through plasmodesmata, through the symplastic route, or through the apoplastic route, which is through the cell walls. And then there's cell surface molecules and receptors. So these are on the cell surface. Like, as we learned in previous units, the glycoproteins of the plasma membranes play a crucial role in cell-to-cell recognition, communication, etc. So these cell surface molecules can interact with each other, which is a form of signaling. And also there's the cell surface receptors, which bind to water-soluble ligands. Now, it's crucial that you remember that cell surface receptors only bind to water-soluble ligands. They cannot bind to fat-soluble ligands because fat-soluble ligands bind to intracellular receptors. Because remember, fat-soluble molecules can cross the plasma membrane, but water-soluble molecules cannot, and so they have to bind to receptors on the cell surface. Now, there's 3 stages of cell signaling. First, there's signal reception, ignore the missing N, but this stage is relatively simple. The signaling molecule or the ligand binds to the receptor either on the cell surface for water-soluble ligands or inside the cell for fat-soluble ligands. And then signal transduction occurs inside the cell. Now, signal transduction is important in water-soluble ligand messaging, but if the signaling molecule is fat-soluble, usually signal transduction does not occur because, as we will explore it later, but yes. Anyway, signal transduction involves the use of relay proteins, which upon binding of the ligand to the receptor, these proteins can become activated by, like phosphorylation, other mechanisms like calcium bonding. And then these relay proteins can activate second messengers, or the ligand binding to the receptor itself can also activate second messengers. And then a phosphorylation cascade is usually involved in signal transduction. This is when proteins in the transduction pathway, they phosphorylate each other, and then phosphorylation can lead to activation of subsequent proteins in the cascade. And signal amplification is when like an activated relay protein or an activated kinase can go on to phosphorylate a large number of different molecules, which can then, amplify the signal. Now, finally, we have the cellular response. So this is what ultimately the ligand binding to the receptor yields. So the response can be mediated in a variety of ways. So there can be like the breakdown or synthesis of various proteins, hormones, etc. And this is usually done by transcriptional gene regulation. So like transcription factors can bind to genes and either inhibit or promote its transcription and translation and then protein production. And it can also yield in cytoskeleton rearrangement. Now, this is crucial in cell division. In prophase, various phosphorylated proteins activate the breakdown of the nuclear lamina. And cell division, apoptosis, those are all part of the cellular response. Now we'll be moving on to the specific transmembrane proteins. So first we have G protein coupled receptors, or GPCRs. These are transmembrane proteins, so they bind to water-soluble ligands. Now, water-soluble ligands, common ones include hormones from the posterior and anterior pituitaries, such as growth hormone, oxytocin, melanocyte stimulating hormone, etc., follicle stimulating hormone, and also insulin and glucagon, as I'm sure a lot of you have heard about. Those are also water-soluble ligands. Now, GPCRs usually come with 7 transmembrane alpha helices. Now, I don't think you have to remember this fact for AP Bio, but if you're interested, then yes. And I think they have 3 different subunits, but I'll have to double check on that. And GPCRs are coupled with a G protein. So GPCRs are the receptors, but then the G proteins are the ones that are being phosphorylated and actually activate the enzymes. So the G protein can act like a GTPase. So when the ligand binds to the GPCR and then the G protein is phosphorylated, it can activate the enzyme, but of course the G protein with the active GTP does not want to be active forever, and there has to be a way to terminate the signal. So G protein can actually dephosphorylate GTP into GDP by itself. So it can act as a GTP ACE. Now remember, ACE is usually like the ending for an enzyme. Now yes, GPCRs bind to water-soluble / polar ligands, and GPCR signaling is usually slower but more long-lasting than ionotropic signaling. Now, ionotropic signaling includes signaling by neurotransmitters, electrical signaling, etc. Next, we have receptor tyrosine kinases. So these are also transmembrane proteins. So they bind to water-soluble ligands. Oh, and by the way, epinephrine and norepinephrine are both water-soluble ligands. And epinephrine and norepinephrine are brought up a lot. So it might be helpful to remember that. And the whole epinephrine pathway in glycogen breakdown. Now, RTKs are usually activated by dimerization. So when 2 receptor tyrosine kinases come together, they're usually activated. And then the tyrosine residues are phosphorylated, which fully activates the receptors. And then now various relay proteins and enzymes can bind to these tyrosine residues and can be phosphorylated or activated, and then go on to activate more proteins and eventually lead to the cellular response. So RTKs allow for great signal amplification because there's so many tyrosine residues, and as opposed to GPCRs, receptor tyrosine kinases they can activate many pathways using the various tyrosine residues because they can phosphorylate various relay proteins. And these are crucial in the growth factor response. So when someone gets injured and they have to replace their epidermal cells, let's say, of the skin, there needs to be rapid signaling, rapid replacement of the skin cells of the damaged area. So RTKs are especially well suited for that. And next we have ion-gated channels. So these are also transmembrane proteins. So yes, the water-soluble ligands bind to it. And ion channels can be either voltage-gated or ligand-gated. Now in ligand-gated channels, a small ligand like a cation or anion, such as sodium in the diagram here, it can bind to the ligand-gated channel, which can either open the channel or close the channel, depending on the channel's nature. And then for voltage-gated, a change in the membrane potential can open or close a channel. And in the diagram, there's also mechanically gated. So when pressure is applied or when pressure is lifted, it can either open or close the gated channel. And then some channels are actually always open. So in maintaining the membrane potential of animal cells, usually there's a few potassium channels that are always open, but sodium channels are usually closed. Now, this is essential for maintaining the animal cell potential, which is usually negative and closer to potential of potassium. And then the ion-gated channels facilitate flow of cations and anions. And yes, they're crucial for the resting membrane potential. And they play huge roles in neuronal signaling and action potentials, generating the action potentials, depolarization, hyperpolarization, etc., which will be discussed further in Campbell Biology chapters 40, 48, 49. And there's a lot of ion-gated channels in the smooth ER and sarcoplasmic reticulum, which is in skeletal muscles. Now, remember, smooth ER actually stores calcium ions. So, ion-gated channels are crucial for the calcium cations to exit out during signaling. And finally, a receptor that is not a transmembrane receptor, we have intracellular receptor proteins, which fat-soluble ligands bind to. So remember, like fat-soluble ligands, they can cross the plasma membrane and then bind to a receptor inside the cell, in the cytoplasm. Now, when the ligand binds to the receptor, it forms a hormone receptor complex. Now, this hormone receptor complex can either be in the cytoplasm and it can migrate to the nucleus, or it might actually be in the nucleus. And then the ligand will go cross the plasma membrane, cross the cytoplasm, and then enter the nucleus and then bind to the receptor there. But ultimately, once inside the nucleus, the hormone receptor complex can activate transcription factors, or it can act as a transcription factor itself, and then stimulate or inhibit the synthesis of various mRNAs, which leads to new proteins, or if transcription is inhibited, then the proteins will not be made. Now, phosphorylation and dephosphorylation are very important in cell signaling. So, protein kinases actually phosphorylate proteins. So, they take a phosphate group from ATP and transfer it to proteins. So then the ATP is hydrolyzed into ADP. And then protein phosphatases, they combat this action by removing phosphate from the proteins and then bringing it back to ADP, which turns ADP back into ATP. So these two counteract the activities of each other. And then phosphorylation can activate or inhibit proteins. Usually this is done through conformational changes, which is like a shape change in the protein. So when a phosphate group is added to a protein, it can change its shape, and in turn, it can boost or inhibit the activity of that protein. And yeah, phosphorylation is extremely important in the phosphorylation cascade, but dephosphorylation is just as important because you don't want the enzymes to be active forever, which will lead to cellular damage due to like an increased signal. Now, second messengers are small molecules that act in signal transduction pathways. So we have cyclic AMP, calcium, and cyclic GMP. Now, you do not have to focus that much on cyclic GMP, but it's nice to know that it's there. So for cyclic AMP, adenylyl cyclase converts ATP to cyclic AMP. Oh, and by the way, AMP stands for adenosine monophosphate. And cyclic AMP phosphodiesterase converts the active cyclic AMP back into just AMP, which is the deactive form. And cyclic AMP has an inverse relationship with glucose. So when cyclic AMP goes down, glucose is abundant. But when glucose levels drop, cyclic AMP is increased. Now, this is extremely important in the lactose operon, or lac operon, which we will discuss further in Unit 6, I believe, Campbell Biology chapter 17 or 18. And then we have calcium. So calcium concentration inside the cell is kept extremely low. So calcium is continuously pumped into organelles such as the smoothie R and sometimes like mitochondria, and it's also pumped outside the cell. And the cell uses ATP powered pumps to keep the intracellular concentration low. And calcium influx acts as a signal. So since the calcium concentration in cells is normally super low, mass influx of calcium can serve as a signal for the cell, which is crucial in muscle contractions, fertilization, and neurotransmitter release. And also calcium can bind to calmodulin, which is a protein. And then, similar to the hormone receptor complex, when calcium binds to calmodulin, it forms a calcium-calmodulin complex, which can then activate calmodulin-dependent protein kinases. Now, we will talk about that later. And then, cyclic guanosine monophosphate is similar to cyclic AMP, but it has a guanosine instead of adenosine. And cyclic GMP is important in phytochrome signaling and light responses in plants. So like in the etiolation, de-etiolation responses. Okay, so cyclic AMP pathway. So first, as always, the ligand binds the receptor, signal reception stage. And then adenylyl cyclase is activated. Oftentimes it's phosphorylated. So adenylyl cyclase is embedded in the plasma membrane. And then adenylyl cyclase converts ATP to cyclic AMP. And then intracellular cyclic AMP concentration rises. And then that cyclic AMP can activate protein kinase A. And then protein kinase A can further phosphorylate additional proteins. And cyclic AMP phosphodiesterase cleaves cyclic AMP back into AMP, its deactivated form, so that the signal is not perpetual, or that it's not endless. So cyclic AMP phosphodiesterase is like always on the hunt for cyclic AMP in the cell. It's always searching to hydrolyze cyclic AMP back into AMP. Okay, so IP3 as a second messenger. IP3 stands for inositol triphosphate. So like the cyclic AMP pathway and many other pathways, the ligand first binds the receptor, that's the signal reception. And phospholipase C cleaves membrane lipid PIP2. I forgot what it stands for, but it's a membrane lipid. And then phospholipase C cleaves that membrane lipid into DAG and IP3. So now DAG can activate protein kinase C. It's not really a direct second messenger because it can just activate the protein kinase, but it can act as a second messenger for other pathways. And then IP3 can actually bind to ligand-gated calcium channels in the endoplasmic reticulum, the smoothie R, and the sarcoplasmic reticulum. And it can also bind to some calcium channels in the mitochondria. And then calcium is released. So then, remember, intracellular calcium is typically extremely low. And then this calcium release can raise the concentration of calcium by like up to 100-fold inside the cell. And this increased calcium can activate protein kinase C, other protein kinases, other enzymes. And calcium can also bind to calmodulin, as I mentioned before, and form that calcium-calmodulin complex, which can activate calmodulin-dependent kinases, leading to more phosphorylation, more of that phosphorylation cascade, more activated proteins. So this is like a bonus. You do not need to know this for AP Bio, but just for you curious minds out there. So this is the arachidonic acid pathway. So similar to the IP3 pathway, another phospholipase known as phospholipase A2 cleaves a membrane lipid into arachidonic acid. Now this is a fatty acid with a 20-carbon backbone. And so when arachidonic acid is cleaved, it can be incorporated into the cyclooxygenase or lipooxygenase pathway. And then the cyclooxygenase pathway yields prostaglandins, thromboxanes, other non-steroidal anti-inflammatory, I forgot what D stands for, but yeah, non-steroidal anti-inflammatory, let's say, molecules. So prostaglandins can dilate blood vessels, lead to inflammation. They're like important for mammary gland contractions. And thromboxanes are important for blood clotting, like with the platelets and all. And then the lipooxygenase pathway yields leukotrienes. So leukotrienes are extremely important in the immune system. So they can recruit other white blood cells to areas of infection or damage. And yeah, they have the inflammatory actions. And then NSAIDs, or non-steroidal anti-inflammatory, this includes ibuprofen, or more commonly known as Advil. It's those anti-inflammatory medicines that you take when you have inflammations. Okay, so that was a lot about signal reception, signal transduction, second messengers, various pathways. But now we'll talk about the actual cellular response. So what happens after all that phosphorylation, activation, etc. So kinases, typically the last kinase in the cascade to be activated, that kinase can activate transcription factors after it enters the nucleus. Now remember, DNA is always in the nucleus, so yeah, the kinases have to actually enter the nucleus to have an effect on gene transcription. Hormone receptor complexes can act as transcription factors themselves and bind to the regulatory sequences on DNA. But yeah, transcription of genes leads to translation and then the production of a protein product. Now, the protein product or its absence is like the end result of a signaling pathway. Like as we saw in epinephrine signaling, when epinephrine binds and then the whole phosphorylation cascade occurs, the last kinase enters the nucleus. And that last kinase binds to a gene regulatory sequence, a promoter, that promotes the transcription of glycogen breakdown proteins so that the cell can synthesize those proteins which break down glycogen, making glucose available for the body. under stressful conditions that require energy. So now we have apoptosis, which is programmed cell death. Now, it sounds bad, like cell death, but it's actually really good. It helps prevent cancer. And this type of cell death actually conserves the cell's components. So like the organelles, yeah, they're like conserved. The cell forms like little blebs, I'd say, like pinchings. And opposite to lysis, it like kind of shrivels down, I guess. And yes, it recycles its organelles, and it minimizes damage to surrounding cells, as opposed to necrosis. So necrosis is a type of cell death that is not good. It's when the cell swells, and this results in the lysis of the cell, and spillage of organelles, spillage of cell components, and damage to nearby cells because of damage or intracellular damage. So apoptosis is responsible for separating the digits or the fingers and toes of humans and related mammals. So when we're born, we actually don't have distinct fingers, but the parts between our fingers, they undergo apoptosis, so the cells between our fingers, they die, and that's how we have like these separated fingers and toes. And caspases are proteases, which are molecules that break down proteins, or enzymes that break down proteins. And then caspases are involved in apoptosis. So in humans, cytochrome C, as you may remember from Unit 3, the cellular respiration electron transport chain, cytochrome C was a key part of that. So cytochrome C is usually located in the mitochondria, but if there's cellular damage, cytochrome C can be released from the mitochondria, which can activate various caspases in the cytoplasm and lead to apoptosis. So in humans, BCL2 inhibits cytochrome C release from the mitochondria. So usually it's suppressed, but when there's cellular damage, BCL2 does not inhibit cytochrome C release, and then cytochrome C can be released, activate caspases, and lead to apoptosis. In C. elegans, BCL2 is kind of replaced by CED9. It's a type of caspase bound to the mitochondria, and this protein usually inhibits CED4 and CED3, which are caspases further along the apoptotic pathway. But when CED9 is not bound to mitochondria, it basically activates CED4 and CED3. The brake pedal is lifted. And then when these caspases are now active, they activate other caspases in the apoptotic pathway and further proteases and nucleases. And then they break down the nuclear lamina, DNA, RNA, proteins, organelles. And this leads to apoptosis. And yeah, that was it for cell communication. So if you have questions, please leave them in the comments below. And I know we didn't dive into the super tiny details, and we skipped over a lot of sections such as water soluble versus fat soluble hormones, like examples of those, and different types of kinases such as Janus kinases. There's a lot to learn about this, so I will be uploading another video that goes into deeper detail past the scope of AP Bio. But yeah, that was all I had today. So please leave questions in the comments. And thank you so much for watching.